protein interaction, active site analysis, and many such work like network pharmacology. At present, Dr. Priya Gahlot Madam is working as Clinical Research Coordinator in Interactive Research School for Health Affairs under the Bharati Vidyapeeth Deemed University, Pune. So, on behalf of All India Institute of Ayurveda and the member of this lecture series, I welcome you all, Madam. So, now the platform is for you.
Hello everyone. Good evening all. I hope I am audible to all.
Yes, Madam.
So I will quickly share my slide with you, everyone. So today we are going to discuss so I will be covering the topic bioinformatics, and as we all know that what is bioinformatics, if we discuss, then it is an interdisciplinary field that basically applies computational methods, and we analyze and interpret vast quantities of the biological data which is available using certain algorithms and softwares. So that study is basically known as bioinformatics. And how we study bioinformatics is by there are certain tools and components to it. There are databases, computational tools, we do data mining, and there is molecular modeling and etc. So there are many tools using all these components. We basically use to analyze and interpret our biological data, and we basically acknowledge.
Dr. Priya, we are unable to hear you.
Wait, sir, I think I'll share my presentation again. I
Yes,
think
madam.
there
Please.
was some glitch in the network. Yeah.
Yes, madam.
Sorry for the inconvenience. I hope now it is visible.
Yes, madam. It is visible. Your voice is also clear,
Yes.
madam.
Okay. Okay. So basically, by using certain computational method, we are analyzing and interpreting our available biological data. So that is bioinformatics. If we move on to that, how we can use the bioinformatics or informatics in Ayurveda, then we can do herb drug discovery by identifying certain active compounds in the medicinal plants using the molecular targets. We can do certain herbal and drug discovery studies. We can do herbal drug interaction studies. We can also find out drug repurposing by using new applications for traditional Ayurvedic formulations, like phytochemical databases studies, which can be developed for Rasayan herbs. Network pharmacology studies, we can study polyherbal formulations using those. Medicinal plant DNA by DNA barcoding. We can do genomic studies by understanding Prakriti and the constitution through genetic variations. We can also do personalized Ayurvedic medicine studies based on the genomic profiles or the available biological genomic data with us. we can do scientific validation of the Ayurvedic principles at the molecular level also. So basically, in my lecture today, I will be covering more of what bioinformatics and how we can I will like to show you and focus more about how bioinformatics can play a certain role in Ayurveda. And I think in our last lecture we have discussed much about the molecular docking simulation and admit studies, so I'll be diverting a little bit and focusing more on these aspects. So, if we talk about the importance in modern Ayurveda practices of bioinformatics, then we can do evidence-based validation using this. We can do drug development acceleration. We can um, safety and standardization, personalized medicine, and global recognition. There are certain biological databases. So, like Gen Bank. Uh, I will like to show you and visit all the websites, and I will like to give you some uh, a feel of how these databases look. So I will quickly like to is my screen visible? I will like to just want to know that if the other screen is visible or just my presentation is visible to everyone.
Your presentation is visible, madam.
Wait a second.
You can minimize. So
Yeah. No, I need to reshare my screen because just a second. Yes. So this is NCBI, and as we have discussed that there are certain databases in NCBI, that is one is GenBank. So what happens with GenBank, I will like to show you. Yes. Uh, Basically, in GenBank, we can search any available nucleotide data. So if you can search, so basically we can extract certain nucleotide data from using GenBank. We can search or we can assign that what kind of filters we want. If we want to study plants associated or playing role in this particular gene with this particular gene data, or you want to specify or focus on just one organism.
Madam, we are unable to hear you. Dr. Priya? Dr. Priya? Dr. Priya, we are unable to hear you.
Hello.
Dr. Priya? Hello? Dr. Priya? Are you
Hello.
able to
Hello.
hear me?
Yes, I
Yes.
think there was certain glitch in between, and
The
I'm really
PPT was
sorry for the inconvenience caused.
yeah.
Uh, I'm
No,
I'm I'm
no, it's okay, madam. Actually,
starting
the
again.
PPT was visible, but we were unable to hear what you were explaining. So you may again
Okay.
share the PPT, and now your voice
Yes.
is very clear, madam.
Okay.
Yes,
Okay.
ma'am. Yes, ma'am.
Okay. So I will quickly share my presentation again with everyone. I'm really sorry for this
It's okay. No, madam. No
uh,
problem, madam. No problem.
connection issue. Yeah. Okay.
Yes, madam. Now the PPT is visible.
Okay. So was like I couldn't know that at which part my voice was not clear and at which part it was.
Yes, madam. This.
So should I start over from the very start or
Yes, madam. Better we start from the beginning.
hmm,
It will be good.
yes. Yes. So today we will be discussing about the bioinformatics basically. So bioinformatics is a science or a very is an interdisciplinary field where we are applying computational methods to analyze and interpret the biological data which is available using certain algorithms and softwares. So for the bioinformatics studies, as we can say, we use certain components that are certain databases. And what are these databases is that they store and organize the biological data which is available to us, like any genomic data, proteomic data, or metabolomic data. So we can have access through using these databases. There are certain computational tools available, like there are certain softwares, there are certain algorithms. Using that, we can analyze our data which is available to us. There is data mining, like we can extract some using certain large biological data sets, we can extract some meaningful analysis to that data. We can also do molecular modeling, like we can predict certain structure and functions of biomolecules, protein structures, and other things. And using those modeling or using those structures, we can further perform other studies as per our interest. So it transforms raw biological data into a actionable knowledge, you can say, for drug discovery, for personalized medicine, and to understand certain mechanistic areas of a certain disease. And if we talk about that how bioinformatics works in Ayurveda, so I guess we have all attended one lecture by Dr. Prashant sir, and he has covered basically a major part about molecular docking, molecular simulation, and ADMET. So today in this lecture, I will be focusing more on how bioinformatics and Ayurveda and how it can play a better role, or what you can do with bioinformatics in Ayurveda. So we can do certain herb drug discovery studies by identifying certain active compounds from a medicinal plant. We can search their targets and where in which disease is playing certain roles. So certain similar studies can be studied or using that model. We can do herb drug interaction studies as like in molecular docking and simulation studies. We can do drug repurposing studies by finding new application of certain traditional formulations, that in if it we can use these drugs in, or we can repurpose these drugs in other diseases. We can do phytochemical databases development for the Rasayan herbs. We can study the network pharmacology models for polyherbal formulations. We can do DNA barcoding for the medicinal plants. We can study genomic studies like to certain, like we can do prakriti through genetic variations. We can do personalized Ayurvedic medicine based on genomic profiles. We can do scientific validation of Ayurvedic principles at molecular level also using bioinformatic tools. So to start with that, and I will like to highlight that to touch certain aspects of bioinformatics in modern Ayurveda practice. How, what is its importance? Like we can do evidence based validation using these tools, which can provide molecular evidence to the traditional Ayurvedic concepts like Tridosha, making them more acceptable to the modern scientific community. We can do drug development acceleration, uh, that can reduce time and cost in identifying bioactive compounds. Using, we can do virtual screening and then we can further do such studies. We can do safety and standardization. We can also study the personalized medicine studies using bioinformatics. We can do global recognition, which can help Ayurveda gain the international acceptance through the uh, scientific data and the other reviewed publications. So this is how bioinformatics can play a certain role in the Ayurveda practices. There are certain, as we have discussed, that there are certain components to bioinformatics, and I will like to cover the basic concepts of bioinformatics. To start with, we should understand that what are the biological databases which are present. So there are certain biological databases like GenBank. We we use it for the nucleotide studies. Basically, there is EMBL, there is PDB for protein structures, there is UniProt for the protein. Any protein you can search its sequences, its functions. It's like there's a whole list of the data available for us at UniProt. There is PubChem for the chemical structures and the certain chemical data set for any chemical compound. So, and what are biological databases? So these are basically the repositories which store the biological information or the data which is available to us, which we can get access. These are freely available databases, so we can access them for our research purposes. So there is one database, as we discussed, that is GenBank. So we can do DNA barcoding using GenBank. We can use GenBank sequences to identify and authenticate Ayurveda herbs using certain genetic markers. There are certain gene annotations, like identifying genes involved in biosynthesis of active compounds, that we can do using GenBank. There is 16 sRNA analysis that also we can do use by using GenBank microbial sequences. We can study gut flora which is influenced by Ayurvedic diet or formulations. We can also do target prediction using GenBank data. So there are certain biological data available of the nucleotide and gene data, and all these databases are also like linked to each other. So the so you can search any of your interest area here, and let's just search insulin. So it will give us everything. Like I had just randomly put insulin. There was no logic behind it. I just wanted to give you that if you will put in a random search to it, I think it is. So you can use this database for studies which are of your interest. Like there is a gene data set for humans. So you can find all the details and all the sequences. So you can perform fast, you can perform blast, you can use this data to like for primer prediction also. You can like, there are many applications for this database, but I will not like to go much deep into it. The next database is EMBL.
Hello, Priya.
Hello, yes.
Uh, can you explain like what is blast, what is whatever you said in short? Because these terminologies are new to Ayurvedic people. So
Okay,
what you said that what is that? So we don't know.
okay, ma'am. Blast is basically a alignment search tool. So we use it in bioinformatics to find similarity between the two biological sequences. So that is BLAST. We can put in certain query sequences, and like there was one sequence. Like you can run BLAST directly from here, so it will search randomly with and will compare it with other. So we need to put in. I have not assigned any particular search to it. I have just to give you what BLAST does. I have run BLAST. So basically BLAST is to actually it's a big sequence, so it will take some time. So I think
So in
this
short,
we can
if
cover later. So I will like to just give you an idea that basically what BLAST does it will compare two sequences, or if we are randomly giving, then it is going to compare that one particular sequence to the whole data set that and will give us the similarity.
so can we compare two genes sequences also in
Yes, we
blast?
can compare two gene sequences also using BLAST tool.
Okay, we can go ahead.
Yes. And then there is this another database, which is commonly known as EMBL. EMBL is basically European Bioinformatics Institute. So we call it EMBL database. So it is a comprehensive nucleotide sequence database, and it is not limited to nucleotide sequences only. You can have multiple analysis tools for sequence alignment, structure prediction, and there is a lot other tools also in EMBL. Like, it is not just about the nucleotide sequence, or you can use just that. There you can predict molecular targets for your herbal compounds using EMBL. You can also model biological pathways influenced by traditional formulations. And you can also explore gene compound interactions using this database. So I'm just telling you that what all you can do. EMBL is a bigger organization, and CAM EMBL, Uniprot, GWAS, Cluster Omega, these are all tools which are available in EMBL. Like, if you will go to EBI, so this is basically EMBL. You can find any gene, any protein. So I have searched randomly keratin. So you can find all the genomes, metagenomes, nucleotide sequences, their structures, protein structures, how they are interacting, their molecular interaction, the diseases where it is linked with. So it will give you the vast data. So databases are where you can find all the data related to that particular search, whatever you want to search. So, like, we can also do P53. So this is basically where it will give you the disease with which P53 is linked with. It will also give you the related genes, where it is expressed, the related proteins, its structures, and the literature, relevant literature for the same. You can also search for the pathways where it is playing role. So this is just what basic EBI search does. But if we look into, the other tools which are available in EMBI. So in EMBI, when we enter, there are certain services which are available for us. So for doing bioinformatics studies, so based on your rationale, based on what you want to do or in which area you want to study, you can, for the GWAS, you can get the GWAS catalog. So it contains all the GWAS data. In identifiers, you will find other things, like
So
in
what is
Uniprot, if we
Jivas?
will say,
Priya, what is Jivas?
I am coming to it, but I will like to start with Uniprot so that it will be easier for all. So Uniprot is basically where you will find all the proteins. It is a protein database. So we can get reviewed data entries here. So it will give you, this is the Uniprot ID. This is the protein name. This is the gene name, its function, its gene anatology. It will give you, like, in what all biological processes this particular protein is playing certain role. Then what are the pathways these are other databases. So whatever database you are using, you can always link certain database to the other database. So they are mostly they are working in collaboration. So they are like, you can correlate them. So here, if you will click this, then it will open another database, which is a pathway comms. So it will give you the pathway search for this particular protein. So it will give you interactions, it will give you enrichment. You can do your pathway analysis studies, other studies. Reactome is also a pathway database. So it will give you Reactome is also a very huge database for pathway studies. So there you will find all the pathways related to your particular protein, where it is playing role, how it is playing role, and then you can use it for the pathway studies. So to just give you a glimpse that CAMI-MBL is for the bioactive molecules, all the chemicals. If you want to search for any chemical, like for a particular plant, you will be required to search certain bioactive molecules which are associated or which are linked to that particular plant. So you can quickly search that in like this is an Indian medicinal plant database. It will give you, like, you can put in any plant name and it will give you all the bioactives. But when you are going to search and when you are going to study the other in detail chemical studies for this, like you can search any compound here. It is a huge data set. So you can visit it and you can put in any chemical name you can think of. This is the structure. These are the canonical smiles, which you can use for your other studies, like in basically molecular docking studies, also for network pharmacology studies, also for virtual screening for your studies, also QSAR studies. You need canonical smiles or NG keys, so you can get that detail from here. You can also find the other details about your this particular bioactive compound. Then there is Jiva's catalog. So it is basically a curated collection of all the published genome-wide associated studies. So Jiva's is basically the full form of Jiva's is genome-wide associated studies. So I will. So here you can like search using any PMID or any particular disease also. So it will give you all the related studies. You can export this data, you can download its association, and then you can do further studies. There is Cluster Omega. It is also a multiple sequence alignment program. In BLAST, you are sequencing, or you are aligning two particular sequences. Using Cluster Omega, you can put many sequences together, and then you can do the sequence alignment of that particular sequence. Using gene 2 phenotype, you can do curation and dissemination of the gene disease association. So basically, it is not a very huge data set, but it is right now having certain limitations to it, but it can provide you data for these particular cancer disorders, cardiac disorders, developmental disorders, ear disorders, eye disorders, skeletal and skin disorders. There is another database that is Swiss Institute for with Swiss Institute of Bioinformatics. You must have heard about Swiss ADME, Swiss Target Prediction, Swiss Doc, or other Swiss tools. So basically, it is a Swiss Institute of Bioinformatics is a institute which is developing, or which is providing services for the bioinformation databases. You can also use competition. You can use these tools. These are freely available tools, and for Swiss ADME, it will give you the admit properties for your, you can do pharmacokinetic studies, you can predict your models, or you can perform admit studies using Swiss ADME. Swiss target prediction will give you that using a certain bioactive small compound, that this compound is linked to or giving similarity index with what all, so the protein and ligand interaction for that studies, you can get some prediction models here. You can use Swiss doc, if you cannot use Autodoc or other tools, Vina, then you can use Swiss doc. It is freely available docking web service provided by Swiss Institute of Bioinformatics, which you can use for docking studies. This also, I can just provide you the so this is for Swiss doc. This is Swiss admin. There is Swiss similarity also, and you can also study, and you can also do protein modeling using Swiss tools. So you can enter your smiles list. You can get the smile list from the CAMI MBL. You can paste it here. And if you don't have smileys for your bio compound, you can also, and you know the structure from some literature database. So you can prepare, like you can draw your bio compound here, and you can get the smileys here. And you can further look into it, that how it is, and what are the admit properties for that particular bioactive. So even though you don't have smileys and you know the structure, then you can get the admit studies for your biocompounds. Then there is Protein Data Bank. So it is basically a global repository for 3D structural data of proteins, nucleic acids, and its complex assemblies. We all know that how complex structures these proteins are. So you can get the 3D coordinates, and you can get the whole data about any protein here. Like you can search your protein on the UniProt, and then you can search that particular 3D structure of that protein, which you can use for your molecular docking studies. You can also check, you can get there are certain uh, criteria to select a protein. So if I just open any random protein, just to show you, there is certain PDB validation tool which is available. So it will give you that what kind of score it is giving. So you can use it in your studies or not. So there is a percentile rank. As per that, you can use that particular protein for your relevant studies. So where you can use this databases in the molecular docking studies and structure activity if you want to know that this particular structure, how it is behaving, and you can understand that particular herbal compound in relation with that particular protein. So this is just an example of the structure of, the insulin like growth factor 1 receptor kinase, which I have just given you. These are the 3D structures which are stored here using experimental methods like X-ray crystallography. Structures are here, NMR structure, cryo-EM structures are here. These databases which we are, the data is stored here, and we are using these data. As per this data, we are using these protein structures in our molecular docking studies and other protein related studies for proteomic studies. And you can get interactive 3D views also for your particular protein, that how it is. Then there is another database which is Binding DB. It is publicly accessible data set base which that can archive experimentally measured binding affinities you can get regarding your protein and ligand interaction, that how similar your particular ligand is. It supports drug discovery, molecular docking, and virtual screening studies by providing quantitative data on the biochemical interactions. There is other database that is DisGeneNet. It is a platform for gene disease association studies, where you can know that if you will put your data, then you will get the large data set that these particular genes are playing role in how many, and in many diseases, how it is relating, and you can provide. Excuse me, I just want to drink water. Like I will take a quick.
Yes, please, madam, no problem.
Yeah. Am I audible now?
Yes, madam.
Yes. So in this GeneNet, basically we put our relevant gene data, and then it will provide us all the links of that where this gene is playing role in how many diseases, and where it is playing role. Like you can get a particular association of your gene and your protein, and how, and in respect to a particular disease. We use this tool for disease biology, system medicine, or translational research studies. There are other important databases also. One is PubChem. It is also for chemical compound studies, similarly as ChemEMBL, as we have discussed earlier. There is one other database, that is Drug Bank. It will give you all the drug information where you can find herb drug interaction details or the data sets. There is CAG. This database you will find metabolic and disease pathways, and you can understand basically the mechanistic of particular disease, and in respect to a particular gene, that where it is playing role and how it is playing role in a particular disease, where in the pathway it is a playing role. There is string. It will give you protein interaction studies. It is used in network pharmacology studies also. CAG is also used in network pharmacology studies. um, PKCSM is a tool for doing admit studies and doing toxicology models. USS safety, toxicity, and interaction of different profiles. Cytoscape is basically a software which will give you interaction networks. So whatever data which you have collected so far from using databases, many literature mining or from anywhere else, uh, that you can basically provide it to Cytoscape, and it will give you different networks, and it will give you link that how a particular plant is linked to a particular disease. There is Autodoc Vina. It is a software we use for molecular docking studies. Also, I have earlier mentioned that there is a freely available tool. Though Autodoc Vina is also freely available for studies, and also SwissDoc is available for freely available for docking studies. You can provide ligand and you can provide the particular protein to it, and then it will give you um, the binding affinity and will give you the molecular docking studies. GROMAX we use for dynamic or molecular dynamic studies for finding that when a particular ligand and binding receptor is binding, then how good they are binding, like what are the stability for that particular binds in a particular simulation models. Then I will like to cover that what is system biology. System biology basically is a holistic approach. So here we are not concentrating on one gene and one disease theory. Biology basically refers to studies for biological systems rather than a particular single system. It integrates and it interacts a network of genes, proteins, metabolites, and isolated components which are available, and it provides a holistic approach. Like there is integration of multi-omics data like gene data, protein data. We are integrating it to system biology. There is computational and mathematical modeling data we do use for system biology. There is network interaction of complex biological systems. So system biology, basically the main principle of system biology is that we are referring to the holistic approach to it. We are integrating different omics. We are using the data from different omics to and providing it to the computational and mathematical modeling, providing network interaction and how all the systems differently are interacting together and are responsible for a particular uh, disease. So both system biology and Ayurveda views human body as a complex and interconnected and a self-regulatory system. So system biology refers to multi-omics profiling, network regulation and feedback, system perturbation analysis, multi-target drug modeling, system resilience modeling. Ayurveda, on the other aspect, like similarly, uh, reflects on Prakriti assessment, Dosh Dhatu, Agni balance, Vikriti concept, polyherbal formulations, Ojas and adaptive strength. And this multi-omics profile and Prakriti assessment are basically providing a convergent theme is the personalized medicine for network regulation and network feedback. And these are related with Dhatu. Dosh and Agni balance as in dynamic homeostasis. The multi-target drug modeling and polyherbal formulation in Ayurveda. This provides synergistic therapeutics. System resilience modeling, Ojas and adaptive strength for restoration of equilibrium. There is other concept which we read about is network pharmacology. Ayurvedic formulation contains many multiple plants with many hundreds of compounds, but how they are working in our system. So, to answer that, we perform network pharmacology answers that that how it is synergistically linked. So there is one Ayurvedic formulation which may contain multiple plants, and that plant or formulation will contain multiple bioactive compounds. These bioactive compounds are linked to multiple molecular targets, and these molecular targets play a role in different biological and molecular pathways which are linked with certain particular diseases. So this particular association or this particular linkage is being explained in network pharmacology. We identify the core targets and hub nodes in different complex disease networks. We provide which provide the synergistic actions of these polyherbal formulation. We use Cytoscape tool. Cytoscape is a software string database for protein-protein interaction modeling, Stitch, and network analyst for network pharmacology studies. This is just an example of how metabolite target pathway interaction network, which was related to T2DM. Here, it is the formulation is Nisham Laki. So there are two plants. This blue and red part, these are different bioactives, and how these bioactives are linked to different molecular targets, and these genes how they are associated with particular disease. So this association and network is being shown using Cytoscape through network pharmacology studies. If we talk about Prakriti and personalized medicine, then the genomic basis of the constitutional types, uh, the individuals have unique prakriti as per their Vat, Pith, and Kapha dominance, which determines certain disease susceptibility and drug response. So we can do genomic studies using the SMP analysis, which shows genetic variation, and we can correlate them with the prakriti types. There is metabolic profiling we can do using certain genomic studies, which can differentiate different prakriti types and can show different distinct metabolic signatures to a particular profile. I will like to end my lecture here. Is there any question?
Yes, madam. The all participants are requested to put their queries in the chat box.
Mhm.
Dr. Supriya Bhalerao, madam, is with us. Madam, would you like to add some points?
Yeah, what I feel that bioinformatics is a huge field and there are so many databases, so many things we can do with it. She has explained like there are so many databases which can identify, which can give you structure of proteins, which can give you where the protein works. But now it is up to us that how we will interpret it and how we will use it for Ayurveda. because the information is readily available, it is freely available, that everything is fine. But how to use that information for Ayurveda and how to make best use of it, so these things which these bioinformatics people say very often and commonly, we have to first like learn those things. And I think from today or tomorrow we are starting the series on this by Dr. Bhavna Prashar and madam and her group. So what is like we have to understand, we have forgotten somewhere like during our Ayurveda education and MD, uh, DMS, MD, but what is RNA, what is DNA, where RNAs are useful, where DNAs are useful, what is gene actually, what is gene sequence, what are nucleotides. So once we understand this whole picture, then I think this bioinformatics will be more clear to us, and then we will be able to use it in a better way for our understanding and our Ayurveda's like exploring Ayurveda in the light of these things. Because though Priya has mentioned that it is a nucleotide database, she has shown that we can do blast. But how that blast is useful for us, that is something which we need to learn, or which we need to understand. And then only this whole thing will be helpful for us or useful for us. This is what I think. And from today or tomorrow, the series you are starting by Dr. Bhavna Prashna madam, certain things of this will get cleared, and that is how we will learn this bioinformatics also in a better way. That is what I feel. So, thank you.
Thank you so much, madam. So, one more request to participants. If any participant wish to ask anything to Priya Gahlot madam or Supriya Bhalerao madam, they can put their queries in the chat box. So, Priya madam, actually we have created Google Classroom for the participants, those who are here. So,
Okay.
if there are some learning resources or any Google Drive link where they can find some more information about that. So,
Yes, I can share my presentation, though I wanted to give basically how to use these databases, but somehow I was not able to cover it because there were certain uh,
technically
glitches
speaking,
also and
yeah.
so but if anyone want to ask some question, then I think I can reply it to them, that if they have any problem using in certain database, then I can give them the direction to how to get use that database and how that database help them in their particular research area which they want to perform. Like if you want to do certain studies using as of now many people are working on network pharmacology studies only basically, but there is more to it. Bioinformatics is not limited to just the molecular docking studies, simulation studies, or network pharmacology studies. You can also perform network pharmacology studies using RNA sequences also, not just following certain single path which we are traditionally using as of now, because there are many databases available. And as in as system biology says, that there is a holistic approach which we keep for open in this research. So you can basically uh, link your research area to other aspects using different databases, which I want to say. So what you want to do with it basically defines that how you can do it. And there are many tools, mostly majorly tools are freely available to you. So and these databases, if we talk about, these are very basic databases, like you can say GenBank. So GenBank is for basically sad as it is for nucleotide searches, but you can do many more other studies also using these databases.
Yes, madam. Thank you, madam. There is one query. about
Yes.
what kind of interdisciplinary collaborations are most essential for Ayurvedic bioinformatics research.
Interdisciplinary collaboration, as in, like, you can collaborate where what you want to do in Ayurveda. Like, if you want to dive into personalized medicine, then there is a different collaboration which will be required. And if, so basically, what is your research area and what is your research question here? That will, then I can, like, basically answer the question better, in a better way. Because for Ayurveda, if we say, then where such studies are carried out. Like, we are also doing certain studies. So basically, what study you want to carry out, then I can tell you that how you can carry it out. So there are many studies which are basically happening everywhere. In all universities, you will find there is a bioinformatics department, and many people nowadays are working on herbal formulations or herbal plants, if not formulations. So there are many studies which are being carried out everywhere.
Yes, madam. Uh, maybe more is if any participant is having a very specific question, madam can answer in a better way. You are asking
Book.
about the books.
So
Yes, madam.
basically, in bioinformatics, it is constant. It is a very dynamic subject. So if you are using certain database also, if you have used this GeneNet 2 3 years back, and now this GeneNet look completely different. So there is no book for using databases, but yes, there is a book, like, you can learn certain languages, you can learn the techniques that how you can use these databases and for your particular research area. But yes, you can refer some book for understanding the basic concepts of bioinformatics, but that any book can do. Molecular docking study is being carried out at your campus. Yes, it is carried out. We have done molecular docking study in Trifla. Are there any online courses to understand and have hands-on training regarding this? Yes. Um, Website for what?
Yeah, the Astha Parmar, if you
Uh,
want to know specific,
Priya,
then yeah.
for this particular question, whether there are any courses online, so can you share some resources, like which courses on Coursera, on EduX, where they
There are many
where
courses,
for
like, if you can do computer aided drug designing course, then you can learn molecular docking and simulation studies also. Virtual screening will be covered there. So there are many different institutes, and all these different institutes have their own set of areas. So you can, yes, use Coursera for the same. You can also use, there are certain online available courses on molecular docking. But if you want to use and if you want to learn the molecular docking as a whole, then you can very well study that how different databases are being studied or used in molecular docking, because there is certain limitation to it. Like, you, if you want to dock one protein and one ligand, if you want to dock many multiple ligands to one protein, or you want to do protein protein docking, so depending on your research area or interest, you can learn.
And there was a question like whether which institutes or where these studies can be carried out. So almost all universities and specifically the pharmacy departments also have a lot of bioinformatics work they do carry out. So you can like connect to nearest pharmacy department. You can reach to a nearest bioinformatics department And yes, at our institute, that is Interactive Research Field for Health Affairs, Bharati Vidyapeeth, Pune, we also do a lot of this kind of work. There are different departments which are working on bioinformatics, not only my department, but there are other departments. So bioinformatics has become now integral part of research. So many people are involved in this kind of researches, and herbal formulation, network pharmacology, you will find many people, especially the pharmacy people, are working in this area.
There's one question that what are the major challenges in integrating Ayurveda data with existing bioinformatics databases? Basically, what kind of Ayurvedic data you want to use, mm, it will define your challenge. Basically, if you want to work on certain formulation, then you can search the linked bioactives using certain quantified studies, quantification studies you can perform. You can know what are the bioactives in your formulation, and then you can perform your studies. So there are challenges there also. So challenges everywhere, if you ask me, but it basically depends that how you want to overcome these challenges. Because in Ayurveda, it is not usually these databases are designed for you can say modern sciences, or the sciences which are linked to, basically you can say modern sciences basically. So but for Ayurvedic researches, you can use this data in a bioinformatics database very well. You just need to change the path. Basically you can follow, like you can take your plant, you can see where these particular plants, what are bioactives linked to it, what are the similarity index you can find. Accordingly you can move forward. And if you are not willing to use certain Ayurvedic formulation, but some Ayurvedic concept you want to use, then also you can like basically define your research area first of all, and then using that like you can do SNP studies also. You can very well do you can use the RNA sequence databases also. So it basically depends on which Ayurveda concept you want to use in bioinformatics to study. How can bioinformatics be applied in the study of Rachna Sharir anatomy in Ayurveda? Anatomy, I guess you can do phylogenetic studies. How bioinformatics is used in radiological study.
That is
Ra- um,
informatics be used for radiological.
okay. Bioinformatics has not much to do in radiological diseases, but if you want to use AI and ML and other things, that you can do for prediction and other research in this area, but not this. You cannot use these databases, but you can certainly, like, not in radiology, but in Ayurveda, one formulation works on multiple diseases, and with bioinformatics, you know by which, yes, you can know it, know this. Using the gene to disease association networks, you can very well frame the mechanistic approach to your Ayurvedic formulation. You can do such model and you can do such studies. Basically, for Ayurveda, you need to design your model. Like, as you can say, there is one concept of personalized medicine, or customized medicine, you can say. So, that you can do. in bioinformatics also. Can the gene toxicity of certain drugs or chemicals be studied through bioinformatics? Yes, it can be studied. Basic requirements to learn bioinformatics. If you can understand science, you can, like, anyone can learn bioinformatics.
Yes, madam. Actually, this is very initial stage of exposure of many of the participants into this field. So the queries are coming of such kinds.
Like, if you like, I can provide you my mail,
Ah, yes, please,
and
madam.
then you can reach out to me regarding some, if you can, whatever databases which we have discussed today. I was also not able to explain you everything in a way I wanted to explain. That was also a issue today. There was certain technical errors also. But if you are using any database, you can visit all the databases which we have discussed today. You can just research a bit. You can, like, expose yourself to these databases, and if you come across or you find it challenging, and if you want to know where you can apply it, then I can answer you the same. Like, if you have certain research area or some curiosity, that where you can use, how you can use this, then we can discuss regarding the same.
So since this is a Samhita Siddhanta department and we are discussing on Samhita Siddhant platform, so real interesting thing would be to have our own data sets also, databases also for certain things. You can see like how they have developed for detailed databases for genes, for nucleotides. Can we have databases in Ayurveda for dosha samsarga? So 62 dosha samsarga. So how uh, they look like, what are the different 62, have we ever written like what are the 62, what are the rasa combination, 62 rasa combination, can we do docking of these two, like a specific rasa combination is useful for one dosha combination. So that would be really ayur informatics, and there, if we should reach towards that, like we should have our databases for our own science, and how we can do it, that is something we should think from, and we should learn from all these databases. Of course these databases we can always use and we can serve some purpose, but we should also look forward that how we can develop our own databases for our own science and how we can use that. So docking cannot be only molecular docking, it can be rasa dosha sannipat combinations also, there also we can do docking. So we can find out many such examples from ayurvedic text, and we can develop something for our own science. So that is a future.
Yes, madam. Thank you so much, madam. I think there are no more queries. So once again, I am on behalf of All India Institute of Ayurveda, I am very much thankful to Priya madam for sharing her valuable times and giving an excellent lecture on this bioinformatics topics. And I'm also thankful to Supriya Bhalorao madam. Madam is with us from the beginning of this lecture series, and madam is coming and guiding us further and giving her more insight on the topic related to the Ayurveda principles. So thank you so much, madam. And from tomorrow we have the session by Dr. Parashar madam. So we will share in the group the exact time and the topic of the session. So with this, we will close the session. Thank you. Thank you, one and all.
Thank you.